Good morning, children. Yeah. So in the last class, we have started already the discussion with respect to the sexual reproduction in the flowering plant. In that one, we have started the introductory part. And also, we have started the discussion about the morphology of the androecium. So until then, we have discussed about the morphology of the androecium. So today, let us discuss the transverse section of Anther. This is a TS, the transverse section of a male reproductive structure of a flowering plant. Yeah, in the left side you can see the structure. If you take a transverse section of the androecium, what are the things you can identify? The filament extended, the part of the region. In the terminal portion you can identify the anther. So as we have discussed in the last class, it is a diptychus in the condition, and there are 4 pollen sacs you can identify. Yeah, so and inside that one pollen grains are present, isn't it? Yes. So the anther, then now let us discuss what is the meaning of the anther. Last class we have already discussed. The anther is a tetragonal structure. Tetra means the terminology. It is consisting of 4 microsporangia. So that is 4 microsporangia located at the corners. That is 2 in each lobe. That's what we have discussed, the terminology diptychus. And the microsporangia develops to pollen sacs. The later, the microsporangia in the later days or the after development, it will be used as to the pollen sac. They extend longitudinally all through the length of an anther and are packed with pollen grains. The pollen grains may be numerous in number. It may be varies from few to thousands together. Yeah. So in this slide, you can identify the structure of a flower. This is a lively structure. In the laboratory, if you take a section like this, you will get it the structure as it is indicated the above portion. And in here the lower structure, this is given in your textbook also. If you see the structure of the microsporangium, yes, a typical microsporangium is near circular in outline. It is surrounded by 4 wall layers. See this structure. If you take a structure in large portion, the outermost layer is known as epidermis. Then after that epidermis, the next layer is known as endothecium. Endo means inside, the thecium means layer. Then after that one, you can identify the middle layers, and after that one inside you can identify the tapetum. The outer 3 layers, it is mainly give protection and helps in the dehiscence of anther to release the pollen grains. So this is very important for the examination also. The repeatedly number of times they have asked already this question. So just like write the structure of the microsporangium, and sometimes explain like that also they can ask. So that you should write this diagram, complete portion, and a structure enlarged with the explanation part. Okay. So then after this one, see, if you see the complete structure of particular pollen sacs, individually, the structure of the microsporangium, whatever we have discussed, the tapetum, which nourishes the developing pollen grains, the cells of the tapetum contains a dense cytoplasm and generally have more than 1 nucleus. So that nucleus are many. Okay, so that's the characteristic features. How to identify exactly? Okay, the tapetum, it's a nutritious layer. Okay, then, and they can ask one more question also. What is the role of the tapetum? or what is the function of tapetum, like that they will ask the question also. So that you should write the tapetum nourishes the developing pollen grains. And in young anther, each microsporangium has sporogenous tissue at center. It consists of compactly arranged, homogeneous, diploid cells. See, what is the meaning of this diploid? Homogeneous means a similar. The diploid means yes, 2n. That is sporogenous cells. Generally, they are called as sporogenous cells. See, if you take only one portion of the structure, and if you see the structure of the pollen rind also, so this will be like this. Yes. After this one, if you see the structure of a particular sporogenous cells, so this will begins with the process is known as microsporogenesis. What is the meaning of the microsporogenesis? Yes, see here it is in detail, easily they have given in the diagram. Actually, so the process will begins. That means from the beginning only, but whenever they undergo, the development stages, the sporogenous cells are microspore. It is commonly known as microspore mother cell. Okay, and this undergoes meiotic division to form the microspore tetrads. So again, the terminology tetrad means 4. Okay, that microspore arranged in a cluster of the 4 cells. So that's why it is called tetrads. And the formation of microspores from a pollen mother cell, it is commonly known as PMC. As I said in the earlier class also, which is undergo meiosis. This process is called microsporogenesis, because the microspore means pollen rind, the genesis is the phenomena. Okay, see here you can see the structure of the pollen rind, the stages of microspore maturing into the pollen rinds, as finally whenever it attains the maturity, it will burst open. That is a maturidized anther which releases huge number of the pollen grains. And as the anthers mature and dehydrate, that means the water content will be periodically it will reduces. The microspore dissociates from each other and develops into pollen grains. One marks questions also, they can ask how this pollen grains will dehydrate. So as the anther matures and dehydrate, the microspores dissociate from each other and develops into pollen grains. You can see the structure also, one by one, the developmental stages of the microspore mature into pollen grains. Yes. And each microsporangium contains thousands of pollen grains. That's what I told you during the part of the evolution. For the purpose of the survival, okay, if they releases only few number, there may be no guarantee of the successful pollination and the fertilization, so that each microsporangium contains thousands of pollen grains. So they are released with the dehiscence of anthers. Okay, and see here a structure you can identify. So the pollen rind, how it will be. The pollen rind is commonly known as microspores. It is commonly known as male gametophyte. If you see the structure, so this is just above the pollen rind structure. They have given the scanning electronic, that means microscopic photographs. They have given the pollen rinds. It is a beauty of our plant kingdom. So based on these structures also, there are different type of the designs they have made the botanist. Okay, so let us discuss the structure of a pollen rind. Yes, keenly observe the structure of a pollen rind. Yes, so here they have given the model structure. So not exactly always like this, but generally the pollen grains are spherical in shape, and it may be size may be varies from 25 to 50. That is, yes, actually it is my mm. It will be varies from different species, one species to another species. Diameter may be varies from one species to another species, because there is no guarantee in the all species should be 25 or 50 like that. It will be varies. Yes. And the cytoplasm is surrounded by a plasma membrane. See here, you can see the structure. The cytoplasm, it is surrounded by plasma membrane. A pollen rind has two-layered wall. It's commonly known as outer exine and inner intine walls. Okay? Yes. See, what is the meaning of exine? It is a hard layer. Actually, the pollen rind is very, delicate structure. It should be protected. For the purpose of this one, it is actually the outer layer, the hard outer layer is made up of sporopollenin. One mark question also they will ask, what is sporopollenin? Like that they can ask the question. Yes, then you should write exine the pollen rind. Okay, the exine is a hard outer layer made up of the highly resistant organic material. It can withstand high temperature and strong acids and alkali also. So enzymes cannot degrade sporopollenin. So I told you again, it is during period of evolution for the purpose of the production. So that then only that pollen rind should be protected, well protected. Yeah. And the exine has aperture, so called germ pores, where the sporopollenin is absent. Yes. See, actually, based on these apertures also, the classification will be done. Those things we will discuss in the competitive, while we are discussing in the competitive classes, I will tell you. So, based on the apertures also. For example, if it will be containing only one pore, then it will be considered monocolpates. If it will be 2, dicolpates. If it will be 3, tricolpate. Or if it will be 4, then it will be tetra. But here in this diagram, you can identify 3 germ pores where the sporopollenin is absent. Okay? Yes, as the pollen grain germinates, the pollen tubes arises from these germ pores only. Yes. And these pollen grains are preserved as fossils due to the presence of these sporopollenins. Yes, I told you this sporopollenin, it is actually, yes, that means highly resistant organic material. It can withstand high temperature and strong acid and alkali. Because of this nature, it cannot be easily degraded. So these pollen grains are preserved as fossils due to the presence of the sporopollenins. Yes. And after this one, if you see the structure of the intine is the inner wall layer of the pollen rind. It is a thin and continuous layer made up of cellulose.